I think it is around 5 o'clock in the morning. No?
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
A very pleasant afternoon to one and all present over here. Respected coordinator, co-coordinator, today's speaker, Dr. Siva S. Panda sir. I, Ms. Deepika Rani Sahu, Assistant Professor, RIPS, on behalf of RIPS, would like to present US's welcome to all on the 10th day of Session 1 of Series 2 Faculty Development Program on Emerging Trends and Drug Design and Development, sponsored by AICTE New Delhi and organized by Roland Institute of Pharmaceutical Sciences, Berhampur, Odisha. Today we have an eminent speaker with us, Dr. Shubha S. Pandashar. Before starting his talk, I would like to introduce Sir among all of us. Sir is currently working as Assistant Professor in Department of Chemistry and Physics in Augusta University, USA. Sir has worked as Senior Scientist in Sigma Aldrich, Miamisburg, Ohio, USA as research, design and synthesis of isotope-labeled compounds of biological interest from 2015 to July 2015. Sir has worked as postdoctoral research associate in Department of Medicine, University of Florida, USA, as a from 2014 to December 2014, February 2014 to December 2014. Sir has also worked as postdoctoral research associate in the Department of Chemistry, University of Florida, from 3 February 2014 to December 2014. Sir has done PhD in medicinal chemistry from University of Delhi, India, in 2006 to 2010. Sir has completed BPharm in pharmaceutical chemistry from Manipal University, India, in 2003 to 2005. Sir has completed his BPharm course from Roland Institute of Pharmaceutical Sciences from 1998 to 2002. Sir has also done his postgraduate diploma in patents law from Nalsa University of Law, Hyderabad, India, from 2007 to 2008. Sir has received many awards and fellowships in his field. He has received Access Research Award from August University in 2018. Sir has received the Best Poster Award in Flowhead 2014. Sir received travel award from BSc attend a conference in USA in 2009. He was selected for University Grant Commission UGC Junior Research Fellowship from 2007. He has awarded scholarship from All India Council for Technical Education in 2003. Sir has qualified in Graduate Aptitude Test in Engineering, that is GATE, in 2003. Sir has organized one-day symposium on medicinal chemistry in drug development at 70th Southeastern Regional Meeting of the American Chemistry Society. He has served as PhD thesis reviewer for Acadia University, Canada, Delhi Technological University, National University of India. Sir is editorial board member of 10 reputed journals like Current of Discovery, Technologies, Green Chemistry, Heterocyclic Communications, etc. Sir also reviewer of journals in more than 30 journals like Scientific Reports, Lectures, European Journal of Medicinal Chemistry, etc. Sir has published 95 peer-reviewed published articles having 1,364 citations with high index 22. Sir has also got 4 patents, 1 PCT and 3 US patents. Sir has invited in 14 lectures. Sir has presented 34 scientific conferences. With this brief introduction, I would like to request Sir to uh, continue with the lecture. Over to you, sir.
Please share your presentation.
Okay, can you all see my screen?
Yes, yes, visible. You can continue.
Okay, so good afternoon, everyone. So today we are going to talk about challenges in drug discovery. Before I start talking about today's talk, so I would like to thank Dr. Ravi Kumar sir, the principal of Roland Institute of Pharmaceutical Sciences. I would also like to thank Dr. Vishwaran. He invited me to present some of my work and about the challenges in drug discovery during this faculty development program. So once we are talking about the challenges in drug discovery, the first things comes in our mind how to discover a new drug. And when you're talking about discovering a new drug for any purposes, then why it is so difficult, why there are a lot of challenges in developing a new drug. So we have a lot of problems around us, but still we are facing a lot of challenges to develop new drugs for those diseases. So when we are talking about the new challenges and how to overcome them, then now what are the new challenges behind developing new drugs? When you're talking about the developing new drugs, there are so many challenges. For example, it takes a years to get a new drug for a specific disease. And in addition to that, we need a lot of resources. We need a lot of money. We need a lot of manpower. We need a lot of equipments and so many other things to overcome that. So we have to be a little bit more smart in terms of technology, in terms of strategy, how to overcome all these hurdles so we can efficiently and effectively develop a new drug for any kind of a disease. And when we're talking about a drug discovery process, now you probably know a little bit about how this drug development process works. So starting from a target identification and a lead discovery to the approval from the FDA to get the drug to the market. And it cost around $1.5 billion to develop a single drug for a specific type of disease. So it says that how difficult and how expensive and how tedious the process of drug development. And when we're talking about the drug development and the process, so the how the discovery cycle works. So as we know, there are lots of compounds that people are synthesizing academically and pharmaceutical industry and research institutions. Now the thing is how to identify which molecule could be used for which type of diseases and how we can do that. So there are a lot of ways to design and develop or to do the biological screening. But at the center of this process, one thing comes to in our mind is the lead compound. So once we know this is the lead compound, and from there we can easily discover a new drug. For example, we all know about the quinine. Quinine is used for malaria. So for more than 200 years, the quinine is used for malaria. So that is the one of the lead compound we can say, because nowadays a lot of synthetic anti-malarial drugs are developed. And you can see the quinine is the basic moiety for most of the newly developed molecule. You can think about mefloquine, you can think about chloroquine and on. So all are based on quinine. So once we got that quinine is our lead compound, even the quinine is also used as a drug, but nowadays because of resistance, quinine is the use of quinine is slowly going down. But still, we got a lead compound from there, and it is more easier to develop new anti-malarial drug based on quinine so that we can achieve the target quicker. So this is how the lead compound is important in the drug development process. And from where we can get the lead compound? The lead compound can be from natural source. Quinine is from the natural source, from cinchona bar. But there are so many natural sources from where we can get the lead compound. What are the other source? Synthetic world. There are a lot of synthetic processes, synthetic uh, uh, molecules are available in these different libraries from where we can get the lead compound. And the third source is the computer aided drug design. If we are not able to get the natural source or synthetic world, then we need to utilize the computational program so that we do not need to spend a lot of time in discovering a lead compound. And once we got a lead compound, what we can do is the lead compound can be optimized, and then we can develop a drug like molecule. So this is how the process works. Now let's go one by one. Why from the nature? Why we need to choose a nature for the lead compound? Because developing a new drug is a complex process. But just think about it. If we do not know, if we do not have a source like quinine for malaria, now think about it, how we can develop a drug for malaria. From where we need to start? What is the starting point? The chloroquine could have not come up. The mefloquine could have not come up if the quinine was not present. So now it is more difficult, more complex process from where to start. So the natural product, a source of nature, is giving a kind of a lead step and first step to develop a new molecule. Time consuming, it's another problem, because as I did mention to you, if we do not know lead compound, if we do not know any references, we are saying, oh, okay, this type of plant material you can take, then you will feel better for some specific type of diseases. Then all this information saves time in the drug development process. Otherwise, if you just think about developing a new molecule from scratch without knowing anything and coming to the market, it may take 15 to 20 years, and then we may or may not be get success also. Because after it come to the market, then we says, oh, there are a lot of toxicity, we need to withdraw from the market. So there are a lot of issues there. So nature is one of the beautiful source, so which can give a lead compound, and we can achieve the developing new drug molecule in a quicker time. And developing a new molecule is also an expensive process. And when we are talking about the expensive process, we cannot cut the cost during the clinical trials because that is for our safety. However, we can reduce the cost while developing the lead molecule. And there, nature can help us to reduce some of the cost over there. And it is also a difficult task to develop or to identify a new chemical entity without having any information. However, when we get some kind of information from nature, it is much more easier to identify or to develop a new chemical entity for any kind of a diseases. And in addition to that, in synthetic process, there is a low success rate. However, if we take the lead compound from a nature source, then it is a high possibilities that we will get success from there. Then what is the other things why we need to look at the different nature as a lead compound? In addition to that, there are plant species is one of the promising area where we can look for new drug like molecules or new lead compound. So there are more than 250,000 species present in this world. And in addition to that, there are only 15% have been screened for different phytochemical investigation. However, only 6% have been biologically screened. So now you think about it, how many still number of plant species are still present which were never investigated. And I'm talking about only plant species. If you think about other natural sources, that means we have a huge opportunity to develop new drug molecules by using the plant sources. Now you may ask, why in that case, why we are not choosing a path to develop a drug like molecules from a nature? The problem is pharmaceutical companies, they do not want to pay attention for the natural sources. Why? Because if, for example, any molecule came or a drug came from the natural source for any life-threatening diseases, it may be from for COVID-19 or it may be for cancer or for any diseases, then that molecule cannot be patented because that is already present in the nature. If that molecule cannot be patented, then the pharmaceutical company cannot get money out of that research, out of that outcome. So this is one of the biggest reason patentability, because of why the pharmaceutical companies, they do not prefer to spend a lot of time on natural products, on natural sources. Okay, so since we are talking about the natural products, since we are talking about the natural sources, and now in this slide, I'm focusing on the plant natural sources. Let's talk about different approaches adopted in developing or identifying new drug candidate from plant sources. The one approach is the random approach. What is that approach? In random approach, the screening of selected compounds like different class of compounds, it could be alkaloids, flavonoids, and so on, to find out a new drug candidate. Or you can just directly screen the plant material, like just biopsy, just take the plant leaf extract, just take the bark extract and do the biopsy. So NCI, that is the National Cancer Institute, utilize this random approach to screen the different plant species. So what they did, this NCI of United States of America, they screened around 35,000 plant species for cancer studies. And they spent around 20 years. They started in 1960 and ended in 1980. They spent 20 years to screen 35,000 plant species. But what they got? They got two drugs, paclitaxel, which is your Taxol, and camptothecin. In camptothecin and paclitaxel are still using as an anti-cancer drug at this point of time. So this is one of the biggest achievement what NCI did. But now think about it. They spent 20 years to screen 35,000 plant species. However, now things has developed a lot. Now we are not in 1960s, we are in 21st century. So the everything is much more developed. If we again go over this kind of process, maybe instead of 20 years, we may do all this screening of this 35,000 plant species maybe in 2 3 years. So this is one of the approach where these two drugs came out by this random approach. The next one is ethnopharmacology approach. In this approach, morphine and berberine, these two drugs were developed during this process of during this ethnopharmacology approach. What does that mean? What is that approach means? This approach essentially depends on empirical experience related to the botanical drugs. That means how the drugs are described, what is the biochemistry, what is the pharmacology of that plant species and all. And based on that, this morphine and berberine were developed by this approach. The next approach is traditional system of medicinal approach, or you can say Unani medicine approach. So in this approach, people were started developing, people were identifying new drug candidates. So artemisinin is isolated from Artemisia alba, which is used for malaria, were identified from this approach. The last approach is zoofarmacognosy approach. This is one of the interesting approach at this point, I believe. So as you know, when we are doing a drug development process, every clinical process starts with testing the drug candidate with animals. So if they got a good success rate with animals, then they go to the human trials. So that means they are getting some kind of sense from the animal studies. Now, if you think about it, in animals, in forest, in a lot of places, animals are also surviving, animals are also getting sick. So how they are getting treated? Because if the animals are not in a specified zoo, so that means the animals do not have any kind of a resource of treatment. So that means the animals are getting treated by themselves. So that means the animals know what to eat when they get some type of sick. If we keep an eye and if we observe the animal activities, then probably we will get a sense if the animal is getting sick, the animal is going and eating some kind of plant or some kind of natural sources to get cure from that problem. So that means if we have a knee observation to the animals, then probably we may get a little bit more idea. At this point, there are not of things are going on in drug discovery process for the zoofarmacology approach, but this is one of the key approach which could be used as a drug development process. Since we are talking about the lead molecules, so there is another thing. So morphine, heroin, codeine were isolated from the poisons like opioids and as a drug candidate. And because of that, it reduces a lot of time to developing a drug molecules for pain management. And here is one more example, which is a teprotide, which is a SE inhibitor, that means angiotensin converting enzyme inhibitor, which is used to reduce the blood pressure. It's used as a drug candidate for the hypertension. And based on this uh, teprotide, they have developed, the synthetic world developed a new drug called captopril, which is an anti-cancer drug. And this teprotide were isolated from snake venom. We cannot get snake venom for treatment of hypertension for everyone. We do not have that much of snake in this world. So in that case, we need to come up with a synthetic molecule. But what is the important thing here? Once we identified the source, the natural source, we got a lead compound from there. And from there, the captopril was synthesized so easily because we used the same fragments where the tetraploid is having. It is much more easier to get that. So you can see this is the tetraploid, the structure of this. If you look at this fragment and if you look at this, the structure of this captopril, they all are similar. So that From there, we got the lead compound, we got the basic things from where to start with. So this is how the natural product helps. Okay, going forward about the drug discovery challenges. So as we know, we have a lot of molecules in this world by now because pharmaceutical industries, academics, places, research institute, they are keep on making different types of molecule in the researches. Now, the next thing is you need to think about your human body because the disease is happening in the human body, and in human body there are a lot of things still we do not know. There are a lot of target sites still we do not know. So now the problem is finding a right molecule for right target. So this is the biggest challenging in drug discovery process. So for that, we need to be a little bit more smarter in developing in a drug like molecules. That's called the rational drug approach. In the rational drug design approach, we can go by this ligand-based drug design method, or we can go by structure-based drug design method. In ligand-based drug design method, we do not know the drug or target structure. That means you do not know in your body which protein is getting affected and what is the structure of the protein. Only what we know the target, the molecule which is used to cure. For example, quinine is used for malaria. When the quinine was developed at that time, or when the quinine was isolated or identified as a drug candidate for malaria, at that time nothing was known about the malaria life cycle. But that was the ligand based drug design. But once we know the structure of the protein which we need to target, then we can design, we can modify the molecule. That's called the structure based drug development method. So let's talk a little bit more insight of that. When we're talking about the ligand based drug design method, so morphine codeine and heroin, if you look at these structures, they all look similar. So when we are talking about the ligand-based drug design method, once we know one drug, now we need to make, or we need to identify, or we need to develop another drug which should have a similar properties than morphine. Now, if you look at this one, morphine, when the codeine and heroin were identified, when they isolated this molecule, they saw the structure of codeine and heroin, which is similar to the morphine, and then they came to a conclusion these molecules could be a good analgesic or a pain management drugs. And fortunately, this codeine and heroin also work kind of similarly how morphine works. Now, since we are talking about the similar studies, so just looking at the similarities between the structure of them, is it good enough? The answer is no. If you look at the first two structures, the banana and the ball, they both are in different structure and they both have different properties. That's understandable. But if you look at the next two structures, the basketball and the orange, they look similar, but still they have different properties. However, the first structure and the third structure, you can see the banana and the orange, they both are different in look, different in size, but the properties are same. So that means just looking up their appearance, just looking the structure is not good enough. We need to look at a little bit more than that. So what are the things we need to look? We need to look the physiochemical properties, the physiochemical parameters, which helps to identify or to see the similarities between these molecules. What are the physiochemical properties? That means we need to look at the hydrophobicity, we need to look at the electronic properties, we need to look at the steric properties. These are the some of the example, but there are a huge number of properties we need to consider when we are looking for the similarity studies. And when we are talking about the studying the similarities properties, it is not easy by just looking the structure we can identify. So here the computational programs comes to the picture. These computational programs helps to consider all these parameters while developing a model. In that model help us to developing a new drug-like molecule. The next approach is the molecular docking. And when we're talking about the molecular docking, so that means we know the structure of the protein. Now we need to have a little bit better idea. How we will get the better idea? By these two approaches, sampling and scoring. What the sampling and scoring means? Sampling means we need to take some of the interior site of the target protein, and we need to have any kind of interaction with any ligand to come up with a scoring, and then from that scoring we will say how that ligand is better binding to the active site. And if you look at this one, so how we can get the sampling process? So if you, this is your if this is your target site, that means you need to take sampling from different parts. So here in this case they have taken the sampling from here, here, and here. So that means they identify 3 target sites in the receptor, and in this 3 target site it could be a hydrogen bond donor, hydrogen bond acceptor, or hydrophobic interactions, and whatever it is. So they identified these 3 components, and then they develop a ligand having these 3 components, and they use that ligand. If this ligand is interacting with these 3 sites and having a better binding, then we probably going to have a better score. And depending on the better score, we coming up, okay, this ligand is having better interaction with the target site. So this is how this molecular docking works. And when we're talking about a little bit more on the pharmacophore side, so if you look at the structure, this is the structure of the morphine. So here in the drug development process, if you look, just thinking about the synthesizing morphine is very difficult because the structure is so complex. So now the question is how to simplify. What are the things we need to keep? What are the things we can eliminate so that the synthetic process will be much more easier, or to developing a new drug molecule much more easier. So for that, the 2D pharmacophore approach is important. So that means we need to identify what are the components of this molecules are essential for their biological properties. Now, if you look at this molecule, the red highlighted part is essential for the biological properties. That means the other than the red highlighted part, we can ignore, we can modify, we can change depending of our synthetic feasibility. So this is how there are the new drug metazocine and levorphanol so came up as a synthetic analog of morphine. And interestingly, these 2 molecules are showing similar biological property as like morphine. However, if you think about a synthetic purpose, a synthetic aspect, or drug laboratory aspect, it is much more easier to develop the metazocine or levorphanol than morphine, because morphine is more complex structure than these 2 structure. So now we have a kind of a clear picture. If we have this red highlighted part of a structure, then probably we will be ended of a drug-like molecule which will show analgesic properties. Now, since we are talking about the 3D pharmacophore, so now as I did mention to you, the red highlighted part, that means we need to have a oxygen, aromatic ring, and a nitrogen with a specific distance and specific angle to develop analgesic molecules. Now, what are these things? That means we need to have a hydrogen bond donor acceptor. We need to have an aromatic group which could participate as a hydrophobic interaction or van der Waals interactions, and we need to have a nitrogen which could be act as an ionic interaction. So now we do not need to have oxygen, we do not need to have an aromatic group, and we do not need to have a nitrogen. We can have anything which has the ability to form, to do the hydrogen bond acceptor and donor property in van der Waals interactions and ionic interactions. Then probably we ended up with a molecule which will show the analgesic property. So this is how the drug development process works. So now once we identify the key components, now we can easily develop new molecule which can show the desired biological properties. So here this is one more example to see the target side, or you can say the sampling. So from here you can see we have a carboxylic group, we have a OH group, and we have a aromatic group. So these are the R group of amino acids. So now since we want to develop a molecules, so we want to develop a molecules like this, which could have a ionic interaction here, or we can say which can interact with the carboxylic group, and we need to have a hydrogen bond acceptor because serine is going to be act as a hydrogen bond donor, and we also need to have some hydrophobic interaction uh, fragment of that molecule so that we can easily interact with the ligand with the active site. So this is how we can develop a pharmacophore, and based on that pharmacophore, we can easily design the ligands for the synthesizing or developing a new drug-like molecule. Okay, so now since we are talking about the active site conformation, we are talking about the pharmacophore development, we are talking about the pharmacophore triangle. So now look at the structure of the dopamine. So now we know all what is dopamine, what is the properties of the dopamine. So now if we want to develop a pharmacophore triangle from this dopamine. So now we know, okay, so we have a OH, which is the hydrogen bond donor or acceptor, and then the OH hydrogen bond donor or acceptor, and the nitrogen here. So this ionic bond, ionic interaction or hydrogen bond donor. But can we have this type of triangle over here? That's the question. Because these are single bonds. So if these are single bonds, so that means they can be easily rotatable. So that means the position of this amino group could be here or could be here. How we will know? We need to use the locked bond approach. If we have this bicyclic system, then these are not single bond. So these are not single bond, then that means they cannot move. So from here, if we have molecule 1 and molecule 2 and do the biological studies, if the molecule 1 is showing biological activity, that means our pharmacophore triangle is going to look like this. If the molecule 2 is showing biological activity, then our pharmacophore triangle is going to look like this. So this is how we need to identify. That's called the lock bonds approach. So let's see how the pharmacophore triangle looks like. So the pharmacophore triangle of dopamine is like this, not like this. So this is how the pharmacophore triangle developed. Once we got this pharmacophore triangle, now it is much more easier if you want to develop any drug candidate which is going to show similar biological properties like dopamine. So if you just maintain the distance and angle, then probably you ended up with a molecule which is having similar biological properties like dopamine. Okay, so since we are keep on talking about the molecular docking, pharmacophore and all, so we need to know also what is the best score, what is the best complex, what is the best interactions. So that means which compound is my compound collection could be a active on receptor A. So if you have a big set of collections, and now if you identify 3 or 4 or 5 molecule which is having a kind of a similar score, and then you need to identify which one is best out of all these 4 by getting a comparing the interactions, comparing the scoring property, and comparing the other interactive properties, and based on that you can come up with a molecule which is having a better score, and then you can go from there for the drug development process. Now we have identified the drug like molecule by this from the natural source or from the synthetic world or for the uh, computational approach. So now we identify, okay, this is the molecule which is having better interactions and better activity, and everything is done. So is this is good enough for the drug development process? The answer is no. Because, for example, if you have pain in your foot and if you want to take a drug on orally, and you know your drug should reach to the foot because you have pain in your foot, not anywhere else. And when you're taking the drug orally, the drug is going everywhere in your body. And when it is going everywhere in your body, that means the drug can go to your brain, the drug can go to your liver, heart, kidney and everywhere. Now you want to get rid of pain from your foot, not affecting other places. That means if your drug is going to affect your kidney, so that means are you willing to take a drug which will cure your pain of your foot but at a cost of your kidney? Then probably not. You will not prefer to have that drug even though the drug is effective. So that means we need to consider some other factors which could which could be a problem in the drug development process. For example, ibuprofen cannot be taken by a pregnant woman, because if ibuprofen is going to be taken by a pregnant woman, that ibuprofen is going to affect the fetus which is present in the mother's womb. So that means we need to think about it. The drug is going to affect to the brain, affect to the fetus, affect to the liver, kidney, and so on. So those, that means there are so many other hurdles to overcome. Not just thinking of biological activity interactions is good enough for drug development process. So we need to think about so many other things while drug development thing. So that means we need to consider that ADME and the toxicity. So that means we need to think about how the drug is absorbed, how the drug is going to be distributed in our body, how the drug is going to be metabolized and excreted from our body. So all these things, in addition to the toxicity effect of the drug to the vital organ. But if you look at, okay, now we overcome all this ADME and the toxicity, and now it's good for a drug candidate. If that is good enough? Unfortunately not, because that's not enough still. Because for example, if you develop a drug which is cost 3 crores to or 5 crores to cure a disease, probably most of the people cannot afford it because of the high expensive. So that means we cannot take it. So that is another problem. And if you are developing a drug which cannot be patentable, that means the pharmaceutical companies are not going to show interest, then that means it is going to be another issue. Or if, for example, okay, you have a drug, but this drug is having stability for 1 day. After 1 day, the drug is going to be de- uh, uh, let's say decomposed. Then that is also not acceptable. So those kind of factors, those kind of problems are still there. For example, at this point of time, when we're talking about the COVID-19 vaccines, some vaccines got emergency approval for this situation, but still there are a lot of hurdles going on. So the vaccines which are available here at United States of America, that vaccines are not feasible in India. Why? Because of some restrictions. So the vaccine came up by the Pfizer. That vaccine need to be stored at negative 70 degrees centigrade. Can we store the vaccine at negative 70 degree at wherever we want to? Because we need a specific equipment, specific facilities to do that. So those are hurdles and hassles when we are talking about the distribution of that vaccines. So we need to consider all of them. So at this point, we are using some other alternative vaccines, even though those are little bit less effective than the one which is more effective, but we are a little bit compromising there because of this hassle. So we need to consider all this process in drug development process, because these are the challenges in drug discovery process. This is not only we need to think about biological properties, activities, or the toxicity. So there are so many other things we need to consider in the drug development process. Okay, so since we are keep on talking about the drug development process, we also need to think about how to optimize the drug, how to modify the drug in terms of synthetic feasibility. As I did mention to you, if the drug is having instability or the drug is having highly toxicity, how we can change, how we can modify in a smarter way or easier way, so that we do not need to spend a lot of time to develop a drug like molecules. So the one of the approach is using bioisosters. So what are the bioisosters? There are several types of bioisosters, just replacing one group or one atoms with another group or another atom to have the molecule with similar properties, but at the same time improve biological properties and reduce the toxicity. So these parameters affected by this bioisosteric replacement, that means it can affect the size, conformation, solubility, hydrophobicity, reactivity, and all. So why we need to do that? We need to do this to have a greater selectivity so that we can reduce side effect, toxicity, and increase stability. But in addition to that, it can be more patentable. That means when you once you change a group of the atoms or so on, then it's possible that the molecule can be patented. That means you will get more attention by these pharmaceutical companies. So here is one of the example, one of the rationale while developing drug molecules by using the bioisoster approach. The first one is ticonazole, which is currently used as antifungal drug for skin infections, skin fungal infections. So now, when we were thinking of a drug for systematic infection, for example, urinary tract infection or some other type of fungal infection happen inside of your body, then we cannot use ticonazole because of poor solubility. It is not absorbed. So to improve that, there are a lot of bioisosteric replacement took place. So now, after that changes, they came up a molecule which is having better solubility. But when they did the biological studies, they came out that there's having higher side effect. So they continued this doing the bioisosteric replacements. So now finally they came up with the drug which is called fluconazole, which is currently available as a drug for systematic antifungal infection drug. And this is also used for the urinary tract infection and so many other fungal infection happens inside our body. And this is less side effect. And now you can see the highlighted part, and based on that, these are the completely bioisosteric replacement approach. So now once we have all these hurdle crossed over, the last thing is the clinical phases. So we need to go for the phase 1 to phase 4 to get the drug completely effectively and safely available to people for any kind of a diseases. So we need to go over these clinical trial phases. Now let's talk about some research project what we used these approaches to develop in some drug like molecules. So first one is the antimicrobial drug candidates. So why antimicrobial? Since there are a lot of diseases are present as a life threatening, why antimicrobial? We have a lot of antibiotics available in this market, but the problem is slowly we are getting resistant to this antibiotics present at this moment. That means the microbe, the bacteria, the all other microorganisms, they're getting resistance for this currently available antibiotics. So in that case, slowly, and it is a assumption or it is a calculation that by 2050, around 100, around 10 million people are going to die because of antimicrobial resistant infection. However, the cancer related death is going to be 8.2 million. So that means this is before it is uh, losing from our hand, or before we are losing the grip, we should think about a better alternative antimicrobial drug candidates for that. So here these are the list of the antibiotics present currently used in this market. So depending on that, so we use this drug is our starting point, and we try to modify by using the conjugate chemistry approach. So here if you look at this one, the red color molecules are the commonly standard drugs, and from there we modified these molecules by using the conjugate chemistry approach, and then we did these biological studies. And when we did these biological studies, and you can see from this data that our molecules and the standard molecules are not having much difference in their biological properties. That means whatever the effort we did not achieve a lot in terms of improving the biological properties. But if you look at this log p value, which is the lipophilicity properties, that got improved from the standard drug to this modified conjugates. So the because the log p values are also a key factor when you're talking about this ADME properties. Okay, so now we wanted to do a little bit more by we wanted to use this introduce this click chemistry approach and develop some molecules so that we can come up with a better. Now we know how to increase the log p values, but now we wanted to do some improved biological properties. So by using this click chemistry approach, so we developed several molecules, we synthesized several molecules, and then once we synthesize several molecules, we screen them. But now you can see this 1, 2, 3, these are the concentration of for the standard drugs, and now these are our molecules. You can see all the molecules lost the activities. The molecules are completely inactive. So that means because of this modification, this ciprofloxacin, norfloxacin, and all these molecules, the antibiotics lost the biological properties. So now we adopted this QSAR, quantitative structural activity relationship approach. And in this approach, we consider all the parameters, not just the size, but the electronic parameters and all the hydrophobic effect and all these parameters the QSR model considered. And then after we develop this model, we use this approach for the synthesized molecule. And then this computational program also confirmed that whatever the molecules we synthesized earlier on, we got the bad results. And this computational program also gave this kind of a similar information. It echo with the observed data. That means the computational program, the QSR model also told us that whatever the molecules you synthesize are bad. So now this is again we took that on as a positive comment criticism that okay, so that means our model is giving a kind of a accuracy or validated our model. So now we use the same model and develop some new molecules by using that model. And then once we develop that molecules, then again we screen the newly synthesized molecule. Now you can see the biological data, and these are the biological data of the standard, and these are the biological data of R. So our molecules are way active than the standard molecules. So now we have a better log p values, and our molecules are much more active than the standard ones. Okay, now coming to the next project, which is anti-inflammatory drug candidate. So as we all know, for when we're talking about the anti-inflammatory things, these all are the drugs currently used for the pain management or for the inflammation, starting from acetaminophen to indomethacin, including the prescription drugs. So what is the problem with these drugs? Since we have a lot of options, so why we need to think about new molecules? The problem with this non-steroidal anti-inflammatory drug is a lot of problems associated to the cardiovascular problems, heart failure, miscarriages, GI issues, and so on. So to overcome this, we used that natural product curcumin, is one of our the lead molecule, because as I did mention to you before, natural products always give a better lead molecules. So we use this lead molecule and utilize our conjugate chemistry approach to synthesize some of these conjugate molecules. And once we synthesize, we use this 2D pharmacophore approach and identified the hydrogen bond donor and hydrogen bond acceptor. And then based on that, we develop synthesize all these conjugates. And then we studied the anti-inflammatory properties. In these anti-inflammatory properties, if you look at this, our molecules, for example, this molecule you can see, so the blue line is talking about this blue line, the light blue line is talking about the effectiveness of the drug after 24 hours. As you know, this ibuprofen is red line is after 4 hours. So that means this is why the ibuprofen is always prescribed to take the ibuprofen every 4 hours to maintain the ceiling effect. That means after 4 hours, the effect of the ibuprofen is decreases. Why? Because probably that is going to be excreted or metabolized in your body. So every 4 hours, you need to take this drug. So, but however, the molecules what we got is even though after 24 hours the activities is intact. So that is one of the better advantage on that. But in addition to that, our molecules are having less ulcer index in compare to the other molecules. So ulcer index is one of the common problem with the non-steroidal anti-inflammatory drugs. So in addition to that, we also studied the splenocyte activity, which is giving you the immunity property of your molecule, because most of the non-steroidal anti-inflammatory drugs decreases your immunity. So this is one of the factor we also need to consider when we are developing a new anti-inflammatory drug candidates, because that is one of the problem associated with this non-steroidal anti-inflammatory drugs. Now coming to the last project, which is the anti-cancer drug candidates. So in the last project we consider to develop some molecules for the breast cancer, because breast cancer is one of the leading cause of this cancer death, and lots of human women are getting affected. In the United States, 1 in 8 women are getting affected by this breast cancer. So what we did is, whatever the molecules we synthesized, because as we know that curcumin is a wonder molecule and this works for so many other problems. So what are the molecules we synthesized for the inflammatory project? We try to screen them for the anti-cancer properties, and we studied their anti-cancer properties. But as for the data here you can see none of them are active for these cancer cells. That means these molecules which are good for anti-inflammatory properties are not for good for these cancer cells, not good for these anti-cancer properties. Okay, so that we thought to modify this molecule. So we use this hybrid conjugate approach. So now we introduce another molecule here. It is called the DCA, this dichloroacetic acid, which is again a molecule which is having some anti-cancer properties. So we designed and we developed this hybrid conjugates by using this conjugate chemistry, and then all these hybrid conjugates again we screen for the breast cancer, and we got a excellent result on the breast cancer. All these molecules what we synthesized, and then since we got this cell line data, then we took these molecules to the animal studies. And in the animal studies, here you can see these are after the treatment of our molecule, and these are the control. The tumor size of these animals are reduced, and in addition to that, this is the body weight, because when an individual is having cancer, the first sign is the individual is going to lose the weight. So we were observing the body weight of the animals. Now you can see the body weight of the animals are remaining constant in our study. So in addition to that, we wanted to validate how this molecule works and how it is going to showing the effect. So recently a publication came up on the curcumin, how this curcumin is working against this cancer, and this says that this is inhibiting this dual specific tyrosine regulated kinase 2 inhibitor. So we just wanted to see the R molecule, which is showing a good anti-cancer properties, are interacting with this target or not, and we use this molecular docking approach. And from this you can see that we did the molecular docking approach, and we got a very similar kind of results what we observed in the biological data. So here the CMC2, which is we got the better biological property, and it here also it gave a better docking studies in compared to the rest of the molecules. However, all the molecules are showing better activity than the parent curcumin and the parent dichloroacetic acid. So these are the approaches we adopted in developing a drug like molecules. And thank you for your attention, and I'll be happy to take your questions.
Thank you, sir, for your nice presentation. So actually, there are so much challenges in drug discovery and development process. So many researchers are facing the same problems in the pharmaceutical industry. In any research institutes, also we are facing same problems. So you have enlightened so many things related to this challenges in drug discovery. Thank you very much for your nice presentation.
Thank you.
Good.
Thank you
Deepika
so much,
ma'am,
sir.
are you there?
Yes, sir. Yes, sir. Thank you so much, sir, for this wonderful presentation and highlighting the challenges in drug discovery processes. Now, I request to our principal and coordinator of this program, Dr. BVV Ravi Kumar, sir, to present an e-certificate of appreciation to Dr. Siva S. Ponder, sir, as a token of love and respect on behalf of entire Roland family. Over to you, sir.
Sir is joining just shortly.
Uh, Thank you, Dr. Shiva, for sparing your valuable time, and also thank you for accepting our invitation. And sorry for bothering you so early in the morning. Uh, and I hope this today you have given information to all the delegates, which will be helpful to them. Thank you once again. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present the CE certificate of appreciation. Please accept it.
Thank you. Thank you for your kind words. Thank you, sir.
Thank you.
Okay, Shiva. Thank you very much.
Thank you and have a good day. Bye.
Bye. Sir, namaste.
Namaste, namaste. Very good afternoon.
Good afternoon, sir. How are you?
Sure, it is just running. Day to day, it is just running.
Sir, thank you for
Just,
accepting our invitation, sir.
I was listening last 15, 20 minutes. Hey, sir,
Sir,
mobile. Hello.
sir.
Yes, sir.
So, I think it's the last part of your program, no?
Yeah, sir.
Uh,
Last and the greatest,
Last time? Okay,
greatest
this is
program.
second. This is the last means second page last or any other third page will be there, no? No.
Yes, sir. Yes, sir.
A third page is there with you. Another page.
Yeah. Second,
Okay, sir.
last,
No problem. Okay. So can we start? No
so we
problem.
start.
Um, yes. Madam,
Yeah.
continue. Deepika
Just a
ma'am.
moment, sir.
Yes.
Okay, sir. Uh, Okay, sir. Dear delegates, welcome you all again to the second session of day 10th of Series 2 online FDP program on emerging trends in drug design and development, sponsored by AICTE New Delhi and organized by Roland Institute of Pharmaceutical Sciences, Barampur, Odisha. Now it's time to hear from our second most eminent speaker, Dr. Santosh Kumar Mahapatra, sir. uh, Today, Sir is going to be delivered his lecture on best practices in research sensitization and its outreach pathways. Before inviting him for his talk, I would take this opportunity to introduce Sir among all of us. Sir is currently working as professor and principal in Institute of Pharmacy and Technology, Talipur, Odisha. Sir has a teaching experience of 27 years. He is also acting as Dean of Pharmacy Department in Biju Patnaik University of Technology, Rourkela, Odisha. Sir is an executive in IND Chemi Health Science Specialities Limited, Mumbai, for 2 years. Sir has received research grants from AICTE New Delhi for Research Promotion Scheme in 2005. Sir is a member secretary in State Board of Pharmacy, Sikkim since 2003. Sir is an expert committee member in Board of Study of North Bengal University for B.Pharm course since 2002. He is an expert committee member of Faculty Council for PG Studies in Science for M.Pharm courses. He is as a selection committee member of Departmental Research Committee of University North Bengal for PhD registration. Sir is a life member of Indian Pharmaceutical Association also. With this brief introduction, I would like to invite Sir on board.
Very good afternoon. Are it's clear to my slide?
Yes, sir. Yes.
Hello. Yes.
Yes, sir. Yes, it clear.
Yes, sir.
Clear. So very good afternoon to all my friends, including coordinator Professor Ravi Kumar and co-coordinator Professor Vishwamohan. So thank you all for creating a very good platform by AICTE sponsored faculty development program online based. And my all friends, our teacher friends now have been highly benefited due to the online mode. And today I think it is the last part of the second page AICTE sponsored program, and we are taking a very good titled Emerging Trend in Drug Design and Development. So last 10 days I think it's almost all my research persons friends completed so many research articles, topics, and the research modes and sessions all. So now the last part, how to develop in front of the society in terms of documentation. Most of my part is the document as because I am the Dean of Faculty of Pharmacy, Patna University Technology, and in my own experience to sharing regarding this. That is best practices in research sensitization and its outreach pathway. Everybody knows whenever a teacher friend is coming to teaching profession, he or she must be knowing, yes, my first passion is teaching. But now, due to the development and onward practice, onward demand of the day now, he has to think the second sensitization pathway, that is only the research. Research is must. And he has to develop in front of his society, profession, how he is, how he will be projected. Just last speaker, Dr. Shiva Panda, what he told, a very good research title, research topic. So this is the basic way. Nobody can think what you are, but everybody will think what is your research, what is your contribution to the society. So only everybody knows, but I am going to just get a little bit memorize things, memory things, and remembering things to all of my friends. At least everybody should think its own way, research sensitization, that is the real way to create an outreach pathway. Everybody knows from the very inception, everybody knows, all my teachers, colleagues, my teachers, everybody knows, yes. So M.L. Shah was creating his father of Indian pharmacy. Now we are here, and as an academician, as a researchers, as a scholar, so everybody at least remembering the person who is creating our pharmacy profession and highlighting now it is the second lead profession in India now. What is that? Research is activity that makes pharmacy a science rather than a technical skill. Academics is separate, research is separate, but my point, my suggestion is, where there is academics, it is just conjunction with research. Where there is research without academic activity, cannot go ahead. So it is a research is the activity that makes pharmacy a science rather than a technical skill. What is that? The goal of research in pharmacy, advancement in pharmacy practice, promoting human health and societal well-being, promoting quality in institution and the community, promoting professional development. So these are four great points, but the basic goals of research for us as we are the basic member, important members of the society, as a professional, you have to know all the things. What is that? The basic characteristics of a successful researcher. Today, everybody knows, and if you feel it so, if you talk with anybody else, if you discuss with any professional, sir, do you know research? Everybody can say, yes, I know everything. Even the DPharm students, they know practical ways. Each and every day I'm facing, each and every segment, every seminar I'm just facing. Yes, can you know research? Everybody, yes, we know research. But research is a topic which definitely gives a greater sympathetic, not sympathetic, it is motivational thoughts. First is self-motivation. It is a self-motivation. At least I have to do. A successful researcher I have to think, attention to details. So half mind, half work, and the second thing, yes, I can manage. No, you cannot say. Excellent time and management skill, strong written and verbal communication skill. Now I can give this thing in this is the fourth most elaborative and very important parameters for us. Strong writing and verbal communication skill which are lacking. As a teacher concern, I cannot say more, but thing is that 20% they're having the lacking on basic strong written and verbal communication is much more important. How to project in front, how to project your researcher's views in front. So this is the most important. Ability to work independently. So each and every time you are thinking, yes, I can do, but my partner is required, my assistant is required. But now I have to think what I have to do. If I will do the best, then the second person coming, yes, he is doing, I am also doing the things. It is a very practical point, practical concept, I can say you. Ability to work independently is the first criteria to go for successful researcher. Second, creativity, curiosity, passions to source the new knowledge. These are four very important parameters for researchers. Keep abreast of scientific and clinical literature. What is the barrier? Everybody can say I can do, but today, tomorrow, day after tomorrow, and a week it is there, it's okay. But after 7 days, the same thing is coming, a decline in the stage due to our mentality. Only because of that, some lacking, some behind the things, barrier, insufficient time because of clinical teaching administrative responsibilities. So whenever a teacher is coming to a research things, then there is a definitely insufficient time. It is a clue. I have no time. All teachers can say, yes, I have no time. The researcher can say, yes, if I will do this thing also, I have no time. So this is the only important, insufficient time because of clinical teaching administrative responsibility. It's a fact I can say. Second, insufficient funding and extremely competitive grant. It is also another practical point I can say. Insufficient funding and extremely competitive grants. It is also not coming in regularly, in relevant with the projects, with the third. Second, inability to generate ideas for relevant research, lack of collaborators or a mentor. So now, whenever taking a person to be a collaborative way, or you can take a person as a mentor for research, definitely some lacking behind it. So that means you have to choose in such a way our collaborator or a mentor very strong, and they have to give a new ideology and at least give each and every time frame support. Second, lack of availability of advanced training opportunities, a very important parameter. Advanced training opportunity, it is a very important parameter because if you are not thinking, if you are not handling the new research guideline, new research instruments, so a person cannot see more in terms of the research. So very, very important. If any scope is there, any Sunday, any holiday, or any scope within 24 hours, at least a person concerned should taking advantages in training opportunity if he or she has. Second, what is the research pathway? Basically, you are having 5 basic pathway for academia, contract research organization, healthcare institutional setting, industry, and government. These are a five factor in India. You can show up academia, top class institution, contract research organization, different research premier research organization, also the healthcare institutional setting, industry. Now many industries are coming off and coming with the specific ideology. Academia industry interaction also developed with a different institution. Even the AICTE also create that this type of platform, and it is a good scope for all researchers. Last one is government sector. So you can add and you can accumulate all the things with the government authorities. What is that? Research career pathway, academia versus industry. It is very comparative statement, and almost all know. My friends, definitely all teachers, definitely knowing everything. Still, I have to forecast very short thoughts and my ideology. Option and choices. What is that? In case of academia, faculty have the opinion and option to select their own research topic, and in case of industry, research is more or less governed by the market demand. Second, spend as much time on the topic as desired. But in case of industry, it is not like that. Specific timeline must be met. So this is the contradictory point in case of here self-individual identity in academia and industry. Second, publication of work. More emphasis on placed on publication, and less emphasis on publication in case of industry due to their guidelines and all. Next, researchers also are encouraged to present their work at symposium and conferences, whereas in case of industry, researchers are recognized based on project management skill and the end product of their researchers. Next, resource funding. Often have to rely on external organization, government for funding, whereas in industry, self-funded, easy access to resources by the directly industry. Now it is very easy for all researchers to take the fund from the industry and go for the different type of research and its own activities. Second, career option. What is that? Faculty may set up their own research laboratory and may also serve as evaluator of research proposal for research funding. That is in case of academia, and in case of industry. See, after a few years of research, pharmaceutical scientists become more involved in management and decision making and spend less time in the lab. So this is the basic difference. Then also your research thoughts ideology will be declining. Ultimately, you don't know anything. What is that? Thought of research. Academics, a teacher having two way, a teacher having two color. One is white and black. Blackboard and white is two color to give a basic research funda, to basic knowledge. Now that two color is not reflected, it is coming under 7 colors. Each and every color will create a different research activities. Search for knowledge, a careful investigation for the search for new facts, and endeavor to understand the unknown, to establish new relations among known facts, extending, correcting, or verifying the existing knowledge. So this is the most important parameters to share for the basic thoughts of research. What is that? Behind all said, as because the last session, it is a documentary session, and I am leading this the document. What is the most important parameter? Your work, or last 4 years work, last 5 years work, you have to document it. That is the real assets of yours. The important thoughts of the faculty is motivation. Before a couple of decades, research was primarily considered as optional. Only some strongly motivated individuals were undertaking research purely for their self-satisfaction. But now, you know, what is that? If you are having no research, your existence is no more. Academics means research. Academics means research. If you are a teacher, you should know research. Degree of research may be different, but you have to know the research. Research upgrades my skill. Knowledge will rectify. Potentiality will increase. Justification enhanced, and classroom teaching will be informative. A teacher can self-analyze if he or she became a man set up of academic research both in one angle. So these 5 points is like a 5 finger to develop like anything. Research upgrade mind skill, knowledge will rectify, potentiality will increase, justification enhance, and classroom teaching will be more informative. It is very, very important and very, very important for all my faculty friends. So what is that? You can say the type of research. Type of research means it is first is fundamental research. Research on the core matter of a discipline, generally concerned with formulation of new theories and concepts. And second, whenever I go for the applied research, researchers which may be of immediate use of their society may be termed as applied research. Researchers in engineering, medical and pharmaceutical sciences, economics will mainly come under the category. So descriptive research in social science and business research quite often prior knowledge or information on a subject is not available. So descriptive means totally quantitative. You take not a single point, it is take a broad point. Next is analytical research. A construct to descriptive research, analytical research is possible only when prior knowledge or information about the subject is available. My point, directly I can emphasize the things, my dear friends, these are going through because all are my definite the guide, maybe research scholars. So at least whenever a person can choose a title. So now you can see within 1 hour, a person creating many titles, many project titles. So my point is that if you compare with all research analysts, all research phenomena, so it is very difficult to choose a title. It will take minimum many months, like literature survey, then you can go for any sorts of project title. So analytical research, in contrast to descriptive research, analytical research is possible when prior knowledge or information about the subject is available. It's very important. Second, conceptual research. A researcher is involved in developing new concept, mostly in the form of theory. And empirical research, based on experience and observation. Sometimes the experimentally found raw data are also taken care of. So these are all the empirical research methods. So what is the approaches? Our approaches mainly 3 different ways. Basically theoretical, experimental and simulation. So the theory is, this approach is hard to pursue. Basically only strong motivated individuals should choose this approach. Very strong. Because theoretical approach, you cannot satisfy to the person concerned. Theoretical research is very hard. But in case of experimental research, this approach is relatively comfortable and high costly approach, as the relevant instruments may be cost extending to several crores. Researchers must develop a skill to handle the instrument. Research quality is considered highly valuable. The requirement is a well-equipped laboratory or access to other laboratory and relevant journals. So these are the most important for experimental research. And in case of simulation, simulation having 2 simplified approaches. Simulation is that, first is that it is very simplified approach. It is theoretical in nature but experimental in spirit. And second is simplified approach. The important parameter is that the researcher develop a computer simulation model and simulates the prior properties and priorities of the extended objects on the computer. So these are very research approaches which is based on modern era. How to write research article? I have seen many segments. So writing is a methodological phenomenon. Writing paper, writing a common articles, writing a research articles is very, very different. So writing the research paper is not so easy what you are thinking. very important point is that single author versus joint author. If single author, yes, you can. Joint author also you can. But thing is that this is a basic academic performance index must be satisfied. If a single author may be tough, but you have to learn more. If a joint author may be simple, but the academic performance index, which is highly essential for our development and all, living. Declining in different, and you cannot give a proper satisfactory phenomena for our development. So writing the reports in parts and preserving the publishing the articles, undoubtedly the most challenging part in the life of the researcher. Entire academic scholarly career largely depends upon this publishing skill. My dear friend, I told you a simple theory. What you are, that is different, but your project, your presentation is much more important than the skill. That is known as IQ skill. So undoubtedly the most challenging part in life of the researcher is that doing research is good, doing all phenomena is good, but entire academic scholarly career largely depends upon this publishing skill. So your publishing skill is much more important than your writing skill, than your research-based time skill. So this is the hard time to all. Why plagiarism? It's a new concept, new thoughts. We are trying 10 years back, that's not like that, but it is a very common and phenomenon where there is a research boundary, there is a gate. That boundary only can emphasize which is coming under and which is not being filtered. So this is the only way to enter through the plagiarism concepts. So publication potential, it's dependent upon its uniqueness and originality. Everybody knows, my dear friends, in today's plagiarism, while nobody can cheat, so the researchers should be careful enough not to copy from elsewhere. This is very important. A good and clear title with an abstract is needed. In abstract, minimum reference to the bibliography is highly essential. Format for publication, everybody knows. If you take a title format, if you take a conceptual format by the different author, by the different publication house, it's mandatory. But a general conceptual thoughts is important for publishing a format for publication. There are three types of publication format. Full-length research article, that is contains the IMRD style of introduction, method, result and discussion. Second one is the short communication style, only required methods, results and discussion. The third one is rapid pattern style, that is rapid pattern is required, the experiments are limited within 1 or 2. So these are the different type of patterns you can accessible in the different journal, national, international, as per your requirement. So how to outline all the things? There must be a planning for every write-up. Data presentation is the ultimate attraction, is the most important characteristics of the researcher. Data presentation is the ultimate attraction, because whenever go for any focus to the data, it must be create the second phase of interest, second phase of innovation. The quoted figure number may not be put in a separate contents column like in a thesis. Next is draft writing. Write fast and then edit. Whereas others are of opinion of doing both at one, go to cut the time span. Draft vision styles are differently followed by the researchers writer. It's very complicated. Draft writing is very complicated, because one draft may not be chosen by other, may not be acceptable by other. But your draft writing is in such a way that means you must be overthrough adaptation by all the researchers. So first write fast, then edit, whereas others are of opinion of doing both at one, go to the cut time span. Next is final checklist. The title, abstract, tables, diagram with numbers must be arranged by now. Make sure that your title and abstract match the whole text. Dear friends, everybody should know that means title and abstract. Really, it is very important because it is the bibliographic thoughts of all research papers, maybe 13 pages, maybe 20 pages, maybe 5 pages, but your title and abstract definitely give the total match of the whole text. Before sending to any journal, you are advised to read again the journal author's instruction. It's very, very important. Next is references not to be ignored. I am telling you, dear friends, references is very, very important parameter. Reference, any reference, any style, you cannot match. So this is must adhering steps to avoid repeat correspondence with the publishers. Each reference must be thoroughly checked for its availability. Also, some good journals see the right placing of reference in the text, however, it can be later justified by the author at the stage of reviewers' comments, compliances. So these are all concepts, my dear friends. By doing good job and by writing skill is a separate job. By doing the good job by your collaborative research pattern, collaborative researchers, but by documentary or by your review articles or by your writing skill, it's definitely separate than your own skill. So this is very, very important. So how to develop the writing skill, and it is a basic pathway how to develop each and every time. Why I am telling you, the practices is much more important. Reviewers' comments, complaints, and proofreading is very important. Case the reviewers passes and solicitors. This is the last stage before publication. If you are a strong and able writers, researchers, then the manuscripts may be accepted in the first submission, but most of the revised submission. Ethics, honesty, and future scope. These are the most important parameter. If you are not worried about, then the software will be blend you right. Adding to your double thoughts, citing several references. This is the most important at the time of writing skill. Second, identifying and promoting best practices for research integrity. My dear friends, research integrity means what? Uphold research integrity with vigilance, professionalism, and collegiality. So what is that? Your identity, your secrecy, and your potentiality give the proper vision to the society. And second is data handling. Manage research data effectively, responsibly, and transparently throughout the research process. This includes providing free and open access to research data, models, and code underlying reported results to the extent possible, consistent with the disciplinary standards, founder requirements, employer policies, and relevant law and regulation, such as those govern intellectual property. These are most important parameters whenever you are forecasting, we are reflecting your research data for the publication. Authorship and communication. Follow general and disciplinary authorship standard when communicating through formal publication. Describe the roles and contribution of all authors. Be transparent when communicating with researchers from other disciplines, policy makers, and the broader public. My dear friends, authorship and communication both are very vital at the time of your research article adherences. Important parameter as a teacher concerned, all teachers are good mentors. All teachers are very good supervisors. So these two, why I'm just collaborating with them. Know your responsibilities as a mentor and supervisor. Know your responsibilities as a mentor and supervisor. Be a helpful, effective mentor and supervisor to early career. Don't say no at the onset. But if you scrutinize from the day, start with research, each and every segment you have to give a mentorship, you give your supervision to your scholar, then you can give a proper research paper and proper research foundation to your scholar. The most important is peer review. Strive to be a fair and effective peer reviewer who provides careful review, maintain confidentiality, and recognize and discloses conflicts of interest. Why it is that? Because you should not give your own interest, disclose conflicts of interest when you do a peer review for any sorts of communication. The important is research compliances. The last end part, that is known as research compliances. Understand and comply with relevant institutional and governmental regulations governing research, including those specific to a given discipline or field. My dear friend, research compliance is a mandatory parameter for all researchers. So understand and comply with relevant institutional and governmental regulations governing research. Then also the compliance should be comply with proper justification. So these are all thoughts and belief to all of my friends. So doing research, thinking research is good, but maintenance research is very, very tough. My last experience, what I feel to say, we all are mentor, we all are good supervisor. Then also, if we can do our best for our research, and if we do our best for ourselves, then we can serve a best research publication, best research review, and best research articles to the society. So this is all about. Thanks, all of my friends. So if any just query, any questions, please. Hello. Hello.
Yes, thank you, sir, for your nice presentation. So you have discussed so many things related to research, how to go for publications. Again, plagiarism is also a most criteria to check all these mistakes carried out by the researchers. So it will be very essential for the research scholars and also the faculty members who are pursuing their PhD programs also. We are very much thankful to you, sir, for your nice presentation.
Thank you, Dr. Vishnu Mohan and Ravi Kumar sir.
Namaskar, sir.
Namaskar, Niranjan sir. Namaskar,
Thank you
namaskar.
very much, sir, for your presentation and giving your valuable time, sir.
Thank you, thanks a lot. Sure, only short because I cannot give more because everybody knows the research, everybody knows the research articles. So just I am giving a just small remembrance, right? So everybody knows everything still.
Yes.
So thank you all, Dr. Ravi Kumar sir, Vishnu Mohan sir, and
Sir, thank you very much, sir.
For a very good program setting, and thank you all for your AICTE sponsor program. Thank you all, sir. Please. So it is the last time. Only I am taking 35 minutes.
But sir, within short period of time,
Sir, actually, my network is not good.
Okay.
Yes,
Sir, your presentation was short but very interesting.
Yes.
Thank you.
You have told about the ethics for guides, which is very
No, we are all teachers, but thing is that it is also very required for responsibilities, because so you cannot say research is not a concept. Research a passion. Research must be your passion. Then also you can develop. So I have last 6 years I have experience, and as a dean, I know what is the going on everywhere, and I have attended many DSC, Dr. Scrutiny Committee and RCU and all. So I really going through many more lackings. So then also, whenever Dr. Vishwaman told me, sir, please give her topic. So at least I have taken only, I can give you 30 minutes, but everybody should know academics. You can take two colors, what I told you, white and black, but it's not like white and black. We have to the 7 colors. So research is much more important than your academics. Then also I can give you my thoughts to all of my friends. Okay. So
Thank you.
thank you all.
Deepika ma'am, ma'am
Yes,
continue.
thank you so much, sir, for this. Yes, sir, yes, sir. Thank you so much, sir, for this insightful presentation and sharing your knowledge with us regarding the research approaches. It's really gonna very helpful for all of us, sir. Now I request our principal and the coordinator of this program, Dr. BDV Odetola, sir, to the e-certificate of appreciation to Dr. To serve as a token of love and respect on behalf of Indian Poland family.
Principal
Thank
sir.
you, Dean sir, for sparing
Thank you.
your
Thanks a
valuable time
lot. Thanks a lot.
in spite
Okay,
of
God
your
bless you
busy
all.
schedule.
Yes, with this, I
Am
conclude
I
and
audible?
yes, no problem.
Hello.
Yes.
Uh, On behalf of
Okay,
Roland
thank
Group of
you.
Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Please accept it.
Okay. Thanks, Dr. Ravi Kumar sir and Vishwaman sir. Nicely organized.
Thank you, sir.
Thanks a lot. Thanks a lot. Please.
Thanks.
Okay.
Thank you, sir. Thank you.
Thanks a lot. Can I leave?
Yes, sir. Yes, sir. So, so you can share your feedback form and registered, sorry, test link to your registered
Program
email
has
ID.
come to an end.
So please go through this. We will provide the e-certificate at the end of series 3 during our April month. Thank you all. Deepika madam, you continue.
Yes, sir. Here the issue of online education program is comes to an end. Finally, I would like to use this opportunity on behalf of the board and all the individuals. Thank you of this program for his emphasis support. Teachers of this program, Dr. Vishnu answer for handling all the criteria of this program. Next, I would like to extend my warm greeting to all the respected eminent speakers, sharing valuable knowledge and sparing their valuable time with us. Next, I want to thank members with unflinching support and donation. Last but not the least, all the of country for their active participation and patience learning. With this warm words and a kind message, we officially conclude this series 2 program. And as we are told, the e-certificates will be provided for registered email ID after completion of series 3, which is going to be in the month of April. Uh, Thank you all.
Okay, madam. Thank you.